Good morning, students. So today's lecture will be talking about allosteric regulation. As we all know, allosteric enzymes are those enzymes which possess an additional site, that is, along with the active site, they do have yet another site which is called as allosteric site. Hence, these enzymes are called allosteric enzymes. I do repeat, allosteric enzymes are those enzymes which possess an additional site along with the other active site called as allosteric site. These allosteric sites are unique locations on the surface of the enzyme molecules which have unique function, and each enzyme can have one or more allosteric site. The term allosteric was introduced by two Nobel laureates, Monod and Jacob, who used this term allosteric to denote those sites on the enzyme which are different from the active site and which do not compete with the molecules binding to the active site, or they do not compete with a substrate binding to the active site but rather constructively influences the enzyme activity. Allosteric enzymes are those enzymes which have an additional binding site, and they do are called effector molecules. And once a molecule binds to this allosteric site, it brings about a conformational change, bringing about a change in the catalytic properties. The effector molecule can be an inhibitor or it can be an activator. So, this allosteric regulation is highly regulated. There are various regulatory measures in our body which control all the processes and respond to various inside and outside environmental changes. Some of the important properties of allosteric enzymes are: allosteric enzymes, they have an additional site other than the active site or other than the substrate binding site, which we call it as the allosteric site. The substrate binding site is known as the C subunit, and this allosteric or the effector binding site is called as the R subunit, where R stands for regulatory subunit. There can be one or more allosteric sites present in the enzyme molecule. And next property is the molecule which is binding to the allosteric site, which is an effector molecule. It can be an activator or it can be an inhibitor. And once this effector molecule binds to the activator site, it changes the conformation of the enzyme. So when we plot a graph of velocity versus substrate concentration, it's an S-shaped curve, or we call it as a hyperbolic curve. So a modulator is a metabolite which, when binds to the allosteric site of an enzyme, it alters the kinetic characteristics. And these modulators can be stimulatory or it can be inhibitory. If the stimulator is same as that of the substrate, then we use the term homotropic. That is, if the stimulator is the substrate by itself, then such regulatory enzymes are called homotropic enzymes. But if the modulator is different, if the stimulator is different from that of the substrate, then we call it as heterotropic enzymes. So some enzymes can have more than one modulators also. And enzymes with several modulators generally have a specific binding site. In contrast, homotropic enzymes, the active site and the allosteric site is the same. For example, enzyme is aspartate transcarbamylase, which has around 12 subunits. So on the screen, you can see a schematic representation of the allosteric enzyme by its, and you can see the shape of the graph. It is a sigmoidal graph. Next, we'll talk about allosteric regulation mechanism. There are two types of regulations, homotropic and heterotropic regulation. So as I've told you, homotropic regulation is the one where the substrate molecule behaves both like a substrate as well as an effector molecule. Example is the binding of oxygen molecule to hemoglobin. Then in heterotropic regulation, when the substrate and the effector molecules are different. Next, we come across a term called allosteric activation and allosteric inhibition. When the inhibitor binds to the enzymes, all the active sites of the protein complex of the enzyme, it too undergoes conformational changes, so the activity of the enzyme decreases. Next, allosteric activation. This is when the activator binds to the allosteric site and increases the function of the active site and thus increases the binding of the substrate molecule. So there are a few differences between the non-regulatory enzymes and the allosteric enzymes. The allosteric enzymes always display a sigmoidal graph, and when we plot a graph of V versus S, it's rather a hyperbolic graph as predicted by Michaelis-Menten equation. Example is an oxygen binding curve to myoglobin, which is hyperbolic. And the binding of enzymes to substrate is analogous. In allosteric enzymes, the binding of the substrate to one active site can affect the properties of other active sites of the enzyme. So, in addition to these properties, the activity of the allosteric enzyme can be altered by regulatory molecules that do bind to these allosteric site. For example, the oxygen binding tendency to the hemoglobin molecule is affected by H plus carbon dioxide and even by 2,3-bisphosphoglycerate. So here on the screen you can see the effect of activators and inhibitors and no modulators on the enzyme activity. Thank you.